Hello everyone, and welcome to our online lecture series for Bio 1. Today we continue with our organ system discussions by looking at one of my favorite organ systems to utilize. I certainly do enjoy doing this a lot. It's a great calming feature at different times. That is, of course, our respiratory system. And as we will discuss in this lecture and the next lecture on the circulatory system, we have to look at how breathing has evolved in different organisms and how it's changed as organisms have become more complex. The basic idea behind the respiratory system is to fuel cellular respiration, which of course, as you remember from this class, takes our oxygen as an input combined with a sugar molecule, glucose, to provide energy for the cell in the mitochondria. As products of this process, CO2 is produced, as is water. With these different reactants and products, we can take a moment to see which of them are our gases and very quickly realize that the respiratory system is exchanging oxygen coming in for CO2 coming out. Now, it does not matter what type of organism you are, the force driving our respiratory system is diffusion. Differential pressures caused by different concentrations of oxygen and carbon dioxide, either in cells, in the bloodstream, or in the air, cause the exchange of these different nutrients. Now, not all organisms need a complete system devoted to this. Because diffusion is the main driving force, some organisms, like this unicellular algae here, can get anywhere between 1 to 5 centimeters across as a single-celled organism and still rely on just diffusion to exchange all of the oxygen and CO2 that it needs. Some organisms, some animals, also don't need a full system devoted to this. They rely on being very thin themselves, like these flatworms or platyhelminthes, platy for short. These organisms, as we'll talk about in Bio 2, have no respiratory system, but instead are thin enough to be able to exchange the oxygen and CO2 that they need directly across their membranes. As animals became more complex, and we won't talk about plants here and their different methods for respiration, but as animals became more complex and needed more oxygen, had higher metabolisms, we started to see different changes in the respiratory systems. The major evolutionary step that we saw in going from some of our invertebrates, like platyhelminthes, and even some of our early vertebrates, was the evolution of gills as a breathing apparatus. Gills are a feature that we find in fish, whereby water, or H2O, carries oxygen to the gills, diffusion exchanges oxygen for CO2, and then, quote-unquote, fresher water with a more stabilized concentration of oxygen gets passed over the gill again. Now, one of the features of gills is that water will hold less dissolved oxygen than air. So these gills, which are primarily a very thin tissue and highly branched, do not do as much oxygen and CO2 exchange as similar organs, lungs, do in the air. To accommodate for this, some of our earliest land vertebrates use their skin as a breathing apparatus as well. These, of course, are our amphibians. Amphibians, just like in the gills, have dense capillary concentrations, meaning that there's just a whole bunch of blood coming up against the surface, and by being close to the surface of the skin, it allows for direct diffusion to occur. So it's not dissimilar from what we see within the flatworm. But of course, our planet has a large number of vertebrates, but an incredibly large number of invertebrates. And before we talk about the complexities that is our respiratory system in mammals, we should take a moment to take a look at the tracheal system of insects. Now, insects are interesting because, well, lots of reasons, but not least of which that they are made of an exoskeleton made of chitin. Chitin is a carbohydrate polymer that provides structural integrity. It's very stiff, giving insects the crunch sound when they are stepped on. And their tracheal system is entirely different from ours, in part because it integrates so well with that exoskeleton. Rather than having a singular mouth to breathe through, this tracheal system has many spherical holes in that chitin exoskeleton. The spherical holes allow for tubes to be laid throughout the body. And the major difference between the tracheal system and the lung system that we have is that in the tracheal system, the blood of insects, or their hemolymph, plays no direct role. Meaning that the oxygen is going to be delivered adjacent to all of the organs and the blood necessary through these tubes made of chitin that attach to our spiracles, which are the openings. And that the blood won't actually be carrying the oxygen around. The oxygen is carrying itself. And as an example of these spiracles on a caterpillar here, you can see these textured little openings that really look very, very different from what we'd expect as a mouth. Or I guess they look kind of similar. But rather than breathing through one single opening at the front of the insect, air is being exchanged at many points, many spiracles, along the axis of this insect, which I think is super cool. Now, one of the benefits of having this tracheal system is that these organisms are actually, that this system is much more efficient than lungs. It takes less energy to breathe via the tracheal system than it does via lungs. And that makes sense, right? With lungs, you have to constantly be pushing and pulling on muscles to expand and contract the lungs. This takes energy, this takes resources. You are able to maintain a higher metabolism, but it costs you more energy. In the case of insects, they get more breathing done just as a byproduct of moving. And this is where the efficiency comes in. The moving itself of an insect will help ventilate spiracles. And in that way, the lungs aren't an additional cost to breathe, but rather just a byproduct of the fact that they have to move. So when a spider is moving around, granted, a spider's not an insect, but a cousin of them, a spider's legs moving actually help improve that ventilation. And in a bee's case, the location of the wings are actually present where they are to help move air over spiracles present in the bee body. And this system is totally different, but still operates on that principle of diffusion. Now finally, we can go to our last major evolutionary step that we're going to discuss in this class, and that being the mammalian system of breathing. The mammalian system is much, much more complex, with having many different organs that provide different functions along the way to help maximize our metabolism, even if it costs us more energy. So let's take a look at all of the components of the human respiratory system. This image taken from your book has the different parts of the journey blanked out that I expect you to be familiar with. The journey for air when it enters your body begins in the nasal cavity. The nasal cavity is the start point and serves a couple of different purposes. First and foremost, the nasal cavity is lined in mucus. That mucus is there to help clean and protect. It does a good job filtering alongside the hairs in the nasal cavity, and also protects the tissues themselves from cold air damage. Because as air is coming through the nose down into the next chamber, all of the air is warmed, humidified, and cleaned. And these three steps are absolutely critical as to what the nasal cavity is doing. Because ultimately, at the very end point of the journey of the air, it is going to be directly adjacent to blood. You do not want that air to be cold. You do not want that air to have a different water concentration or a low concentration than is expected, because it will pull water out of your bloodstream. And last but not least, you most certainly do not want any particulate. No heavy metals in the air, nothing that would cause damage to the tissue. Therefore, the nasal cavity is there to warm things up, keep them wet, and keep them clean. After the nasal cavity, the air will contact your pharynx, which you may be more familiar with as your throat. The pharynx has a series of flaps beneath it called the larynx. The larynx is your voice box. So it is air passing through these flaps that allow for words to form, and those words are things that you are listening to currently. And after the larynx, beneath the larynx, we have the major breathing tube. The trachea, similar to the tracheal system that we have in insects, because this is the major pipe that is transporting air into our breathing apparatus, our lungs. Now, the trachea itself is quite an interesting and complex organ. The trachea is lined in two different cell types: goblet cells and ciliated epithelia. The ciliated epithelial cells are used to propel mucus upwards to the throat. Now that's kind of interesting. Rather than mucus traveling down with gravity, the ciliated epithelial cells are constantly pushing it up. You do not want to accumulate this dirty mucus down in the lungs. So instead, the trachea is constantly pushing it up and up. Now, ultimately, those ciliated epithelial cells will push the mucus up away from the trachea and into the esophagus. Most of that dirty mucus is actually cleaned, quote unquote, by being thrown into the digestive system. So you are eating a whole lot of this mucus. Now, occasionally, there is so much mucus that is generated and needs to be removed that rather than throwing it down your esophagus, your body finds it overwhelming and will instead instigate you to, quote unquote, hawk a loogie. And that's exactly what you're doing. When you are clearing your throat, it is because there's so much mucus propelled up by the ciliated epithelia lining the trachea that they now need to propel that material out. So rather than eating it, you spit it out. But please don't forget, you do eat a large portion of the mucus that is used to clean out the air that you breathe. The other feature that is quite particular to the trachea is the cartilage. Now, the cartilage is a series of rings on the outside, a one-sided ring, facing the outside of the organism, or the ventral side of the organism, rather than the interior, because under no condition do you want your trachea to collapse. There's not going to be anything that puts pressure on the trachea from the backside, from the interior, because your spine is there. And if something has penetrated your spine, you have bigger problems. But, a karate chop to the throat could potentially collapse the trachea. To prevent this, the cartilage is there as a strong support to help prevent that from happening. Now, the cartilage is supported by smooth muscle that is going to contract to push the air out and relax to help breathe the air in. So the smooth muscle is helping with volume control, while the cartilage is providing physical support. And now, because we've talked so much about the mucus here, I don't want to leave the trachea without talking about what exactly mucus is. Mucus is a combination of three things: water, you do notice that it's very liquidy, the glycoprotein mucin, which is a sugary protein, and salt. This combination of a sticky, syrupy protein and water helps mucus retain particulate from the air. Salt helps it balance out and help keep that environment sterile. Because obviously you don't want something filled with sugar and protein just to serve as a backbone for bacteria to grow. The salt helps keep it sterile. And ultimately, mucus is doing its work to clean air that's entering your lungs. Now, once air enters into the lungs, it's going to move from bigger to smaller and smaller and smaller passages. These smaller passages do have technical names that I don't expect you to keep track of, like the primary bronchus, the secondary bronchus, the tertiary bronchus, and then the bronchiole, and then finally the terminal bronchiole. Each of these different bronchi are just more and more minor tubes that air is passing through. Once you finally reach where the air contacts the blood, you have the alveolar sac. And this is the functional unit of the lung. It is in the alveolar sac that we have pulmonary arteries bringing blood that has no oxygen, and pulmonary veins bringing blood that has oxygen. Contrary to most of your arteries and veins, the arteries that move within the lungs carry high amounts of CO2, and the veins that move within the lungs carry high amounts of oxygen. Now, these alveolar sacs in the respiratory bronchioles are incredibly small, microscopic in fact. They are less than half a millimeter in diameter, and there is no cartilage giving this structure its shape. Instead, this structure relies on air to give it shape. And this air and the other substances that help line this alveolar sac actually drive a number of respiratory diseases that we'll talk about at the end of the lecture. Around these alveolar sacs, you will have tiny capillaries whereby the air exchange is actually happening, because capillaries are small enough to carry single red blood cells through at a time. And then at the very base of your lungs, you have the muscle that's going to be driving all of this movement. That muscle is a diaphragm. Ultimately, your lungs are operating on something called negative pressure. The size of your lungs changes in order to pull air in or push air out. There is no vacuum causing your lungs to be filled with air. It is instead changes in volume. The lungs themselves are maximized for surface area, because just like how we saw diffusion governing the respiratory processes of either unicellular algae or flatworms, diffusion is also governing our processes in our lungs. To increase the amount of diffusion occurring, we're going to maximize surface area. Each of these respiratory bronchioles and alveolar sacs that do look kind of like grapes on a vine have an incredible amount of surface area for gas exchange in those capillaries. In fact, it's estimated that your lungs have something like 75 meters squared in surface area. Considering that 6 feet tall is something like 2 meters, that's an incredible amount of surface area for such a small organ. A couple other terms that are discussed in your book, and your book has too many of these, so I'll go over the 3 that I expect you to keep track of, are partial pressure, which is going to determine the rate of diffusion. And the way partial pressure has this effect on determining the rate of diffusion is because it is our concentration of gases in the air. So this literally measures how much oxygen is in the air, how much CO2 is in the air, and therefore how quickly are we exchanging oxygen and CO2, and therefore how much oxygen can we get into our bloodstream, and how fast can we get the CO2 out. We also have tidal volume, which measures the amount of gas inhaled. And then VO2 max, which is the maximum amount of oxygen consumption that you can perform during exercise. Consumption. The reason that I want y'all to be familiar with these is that partial pressure is not something that you can control. This is something that the environment around us has. But your own health can change your tidal volume and your VO2 max. So when we talk about respiratory health, we're oftentimes looking at how much air is brought in and how much oxygen can be exchanged, or that VO2 max. Now, VO2 max is both a product of the tidal volume as well as the strength of your circulatory system. I was part of a kind of cool study whereby I got to measure my VO2 max. Usually this is a metric that's not kept track of for most people because it's maybe just important to athletes to look at how their air exchange is occurring. But in a biological study looking at how a particular additive might have impacted healing, I got to measure my VO2 max by having lots of different machines strapped to my face and then being told to run as fast as I can until I could breathe no more. Because that's what it takes to measure your VO2 max. To get to your maximum oxygen consumption, you have to run till you almost vomit. It was pretty fun, but not something that I'm going to repeat in the near future. Now, these are all the terms surrounding gas exchange. Because from our discussion of gas exchange, I want to lead to our discussion of respiratory diseases. Now, respiratory diseases are ways in which your lungs are unable to deliver the amount of oxygen necessary for your metabolism. That amount of oxygen depends very heavily on this graph here, called the oxygen dissociation curve. Because this curve goes to quantify how much oxygen and hemoglobin, the protein that binds oxygen, more on that in a little bit, how much they are, or how quickly they are, binding to one another and breaking off. The oxygen dissociation curve has the concentration of oxygen in the air on the x-axis and the percent saturation, or how much binds to hemoglobin, on the y-axis. So very high binding, very low bonding. And as you'd expect, if there's very little oxygen in the air, you're getting very little oxygen binding hemoglobin. As you increase the amount of oxygen in the air, it is not quite a linear relationship, meaning that more oxygen in the air past a certain point has diminishing returns. But ultimately, there is a threshold that leaves this inflection point. Now, this is actually what a lot of oxygen machines for folks who need them, or ventilators for folks who need them, are doing. They're changing the amount of oxygen concentration so that way, if in case there are reasons that your circulation isn't working as well, or that you have problems with blood or hemoglobin, you are still getting as much O2 as you need to drive your metabolism. There are other ways in which we can change hemoglobin oxygen affinity. We can induce a higher affinity, or a left shift, by reducing the amount of CO2 in the air, by increasing the pH of the blood, or by having lower temperatures. In contrast, you can cause a reduced hemoglobin oxygen affinity, or a right shift, by having more CO2 in the air, a lower pH, and a higher temperature. And the question at the bottom of this visual connection plot from your book is quite relevant here. It asks, the kidneys are responsible for removing excess H+ ions from the blood. If your kidneys fail, how does that impact your body's ability to breathe? Well, a high H+ concentration causes your blood to be more acidic. Acidic pH means that you are at a pH of 7 or lower. If you're getting to be lower in your pH, you're actually reducing the affinity of hemoglobin to oxygen, meaning that you are making it more difficult to breathe because your kidneys are failing. That's a pretty crazy stretch. Or not a stretch, rather, but a pretty crazy observation. You have these very distant or downstream effects caused by organ systems that are not at all related directly to breathing that can still impact the conditions of the blood that will then change your ability to breathe, or how much oxygen delivery is happening per breath. It's pretty crazy, I think. Now, real quick before we return back to our respiratory diseases, let's take a moment here to talk about hemoglobin, that very, very important protein that grabs oxygen from the air for us. Now, hemoglobin, as visualized here, is a structure made of, is a quaternary structure protein rather. That means that it is made of several tertiary amino acid chains. And in this case, there are 4 subgroups. Those subgroups are 2 alphas and 2 beta subunits. And each of them is going to surround what is called a heme group. The heme group is actually what's grabbing oxygen. Now that heme group contains iron. Hence, why we oftentimes associate blood, or red blood cells at least, with iron concentrations. Because after all, the hemoglobin's ability to grab oxygen is determined, or it is in part stemming from that ability of iron to temporarily associate with oxygen. It's very critical. Now, as each hemoglobin is grabbing oxygen, we see changes in shape. Hemoglobin is actually more quick to grab the first. And that ease in which it associates with the first oxygen is much easier than the second and the third. And it's much more difficult, it's the hardest, to grab the fourth. So oftentimes, actually, your hemoglobins are associated with only 3 of the 4 oxygens that they can grab. Now, a little aside here is that you can get mutations in your hemoglobin subgroups that cause those subgroups to be better at grabbing oxygen with lower pressure. The ability to grab oxygen is called oxygen affinity. You can get better oxygen affinity at lower partial pressures. Now, an advantage when there's less O2 in the air means that it's somewhat of a disadvantage when there's more O2 in the air, but there are regions on this planet wherein you might think that organisms may want to adapt to less oxygen in the air. So where do we see less O2 partial pressure? Well, at high altitude. And now, there are human populations who have a mutation in a gene called endothelial PAS domain containing protein 1, or more simply, EPAS1, that allows them to grab more oxygen out of the air when there's less of it, driving that association between hemoglobin and oxygen itself. Now, this is a high altitude adaptation that we actually inherit from not Homo sapiens, but our Denisovan archaic hominids, and is known to confer increased athletic performance in some individuals. Therefore, you may have heard EPAS1 referred to as the super athlete gene. it's interesting to think about that this gene is not a Homo sapiens trait, but rather a Denisovan trait. More on that in Bio 2 and about our mixed ancestry as both Homo sapiens and Denisovans and Neanderthals. But moving back to our discussion of hemoglobin. We know that it's made of 4 subgroups. We know that it contains iron. We know that the heme group is surrounded by those 4 subgroups and associates with that iron. We know that we can change the association between hemoglobin and oxygen by changing concentration in the air, as well as by altering the CO2 concentration and pH in the blood and temperature. So let's talk about how hemoglobin works in our bloodstream. Hemoglobin itself is about 96% of the dry weight of our blood. Meaning that most of your red blood cells are actually just giant bags of hemoglobin. Hemoglobin itself is made in the cytoplasm. So in contrast to our endocrine lecture before, whereby all of those protein hormones are synthesized in the endoplasmic reticulum, the rough endoplasmic reticulum, are transported through vesicles, packaged and prepped in the Golgi, and then secreted. Hemoglobin is a protein that's made entirely within the cytoplasm and those free-floating ribosomes within the cytoplasm. Now, hemoglobin also gives blood its color. When the blood has a high concentration of oxygen, the blood will look bright red because of the presence of the iron. Now, when the blood has low concentration of oxygen, the blood will look dark red. At no point will your blood be blue. So what the heck is going on with my veins? Well, your veins are not blue either. Instead, they appear blue to our eyes because the color red, the wavelength of light that is red, does not penetrate the skin very well. It's a lower energy light. The red gets absorbed by the skin and the blue continues through. Hence, your veins will look blue even if they are not. And of course, you know the other association with blood, because anytime you've had a cut, you notice that it is quite bright red. Even if it was a vein that was cut, the moment that blood is exposed to the air around you, it's going to absorb a lot of oxygen. Hence, any blood that you are bleeding will be bright red, as long as it's still alive. When hemoglobin binds to CO2, because hemoglobin is also essential for the export of carbon dioxide, that hemoglobin will turn into something called carbaminohemoglobin. Which is a structural and slight chemical change of hemoglobin, so that way it can bind CO2. And this carbaminohemoglobin is actually the second most common way by which CO2 is expelled from your body. The first and most common way for CO2 to leave your body is actually as a pH buffer called bicarbonate. And this takes care of approximately 85% of the CO2 produced through cellular respiration. Bicarbonate is great because it acts as a pH buffer rather than changing the pH of the bloodstream. We know that changes to the pH can actually be quite bad, particularly those that cause our blood to be more acidic. So it's really good that most of the CO2 that we're breathing out is breathed out, dissolved in our blood as a pH buffer. The last and least common is where the CO2 itself is dissolved in the plasma. And about 10% of your blood or your CO2 is expelled as carbaminohemoglobin, and then the remaining 5% is what's being expelled as dissolved in the plasma. And now CO2 actually it's more soluble in blood than oxygen, so it's being dissolved in the plasma and replacing that oxygen. And the oxygen is instead hopefully being carried around by the hemoglobin. So now that we kind of understand hemoglobin, the carrier of oxygen and exporter of CO2 in some cases, we can return to our discussion of partial pressures and tidal volume and wrap this lecture up with a brief discussion of respiratory diseases. And now, a few more words that will help us in our discussion of respiratory diseases. The first is something called a surfactant. A surfactant is a mix of lipids and proteins that reduce the surface tension at the alveoli, preventing its collapse. And this stems back to my earlier comment that these alveolar sacs don't have cartilage keeping them up, they just have a solution that helps keep that surface tension, keep it bubbly and open. Now, that surfactant is incredibly important, and babies can actually make too little of it early on in childhood, when they're very young. When babies make too little surfactant, it leads us to our first respiratory disease, or respiratory distress syndrome. When you make too little surfactant, alveoli will collapse and babies will not be able to exchange gas effectively. This causes them distress and pain and crying. So, we now have modern medical solutions to help address the collapse of those alveoli and help babies produce enough surfactant to avoid this. Two other terms that will lead us to our obstructive respiratory diseases are the compliance of your lungs, which is essentially a measure of the elasticity, and resistance in your lungs. How much of an obstruction is there in your airwaves? Now, the compliance and resistance are two terms. I'll put stars next to them because they are associated with tidal volume and VO2 max. Oops. Wrong space. Sorry about that. We'll scroll down to where I have my images of different blood-associated diseases. These are not our hemoglobin-associated diseases, but rather our obstructive respiratory diseases. And these are ones in which you actually increase the resistance in your lungs to the point where not enough oxygen is being absorbed by the hemoglobin and transported to your body. These include things like emphysema, the alveoli walls can be destroyed by this, and when the walls of those alveoli are destroyed, the compliance actually goes up. The elasticity goes up. Because they are more elastic, they will expand too far and then they will collapse. And when they collapse, the air becomes trapped. And air exchange is not occurring. One of the leading causes of emphysema is tobacco use. Do not destroy the walls of your alveoli. Another obstructive disease is asthma. Something that my doctors have been trying to diagnose me with since I was young, because I had incredibly bad allergies. Asthma is an inflammation of the airwaves. When the inflammation puts too much pressure or closes up the airways too much, you get an increase in resistance. And therefore, you're, again, not exchanging air as quickly as you need. Last but not least, we have an edema, which is fluid in the lungs. Fluids at the bottom of the lungs, just like mucus accumulating at the bottom of the lungs, are bad. You don't want anything getting to those regions where you're trying to exchange air, oxygen air, and CO2 air down in the alveolar sacs. Edemas will block and fill those sacs, and as we talked about at the start of the lecture, fluids do not hold as much oxygen as air does, and therefore you again have an obstructive disease, in this case because of water or some sort of other fluid being stored there. Now, of course, fluid in the lungs can lead to other illnesses, accumulation of bacteria or other pathogens that will grow in those fluids. Outside of your obstructive diseases, you can have your hemoglobin diseases. Just like we saw improvements in hemoglobin, or situational improvements in hemoglobin with EPAS1, we also have slumps in, or reductions in the association between hemoglobin and oxygen due to mutations, either in the hemoglobin or the structure of the cell entirely. The first hemoglobin disease is sickle cell disease. In sickle cell disease, we change shape of the red blood cells. From being a nice disc here, this cross-section of a red blood cell shows that we have a disc, a concave, a biconcave disc, to maximize the surface area of the red blood cell, that then will change into a crescent because the abnormal hemoglobin forms strands in the cell that then cause the cell itself to extend. This crescent shape can cause blockages, and there are ways in which we think that this crescent shape might help actually combat certain diseases. So this explains why hemoglobin persists in human populations. But the change in shape does reduce the surface area, and as we started with this lecture, we know that surface area is incredibly important in the diffusion of oxygen. Now, last but not least, in terms of our diseases, we have thalassemia. Oops. Which is a mutation that makes our hemoglobin subunits less likely to grab oxygen. Now, when you decrease that oxygen association, you change that dissociation curve and the amount of oxygen that's getting to your body, and therefore you have a type of anemia. And an anemia is generally any condition in which you lack enough healthy red blood cells to carry an appropriate amount of oxygen to your body's tissues. This image is of a person, red blood cells from a person with thalassemia, because their red blood cells are not grabbing as much oxygen as they should be. This individual has thalassemia, and this individual does not. You'll notice a massive difference in the color of their hands here. Again, due to the amount of oxygen being grabbed by those red blood cells. Now, that's where we'll end this lecture, having, crescent shaped. Having discussed a couple of different hemoglobin-based diseases or obstructive diseases. I do want to leave you with some thoughts as to how breathing might differ in different animals, because we will talk about the diversity of life in bio too, so please do keep track of that and how ultimately diffusion and our organ systems vary across different species, whether they be algae, flatworms, bees, or mammals, and how that causes changes in metabolism, or the metabolism that can be supported, and ultimately changes in the behaviors of different species. But for the purposes of Bio 1, please know the organs involved in the respiratory system, how hemoglobin works, and when our respiratory system fails through different respiratory diseases.